Hello and welcome to the Hospital Near Me Medical Career Training Channel. Welcome to our journey through the captivating world of biology. In this video, we'll uncover the mysteries of various biological processes that contribute to our survival and well-being. Today, we are working on the Medical College Admissions Test, or MCAT, to get you into medical school. Today's featured hospital is Good Samaritan University Hospital. It is located at 1000 Montauk Highway, West Islip, NY 11795. Their phone number is 631-376-3000. Good Samaritan Hospital won Best Hospital in Obstetrics and Gynecology 2023 to 2024. Do you need to talk to someone? Get confidential help through talking with a full-services professional listening company. Reduce your stress and increase your peace. Get problem-solving help today. Click on the link in the description that says, "Let me help you in life." Increase your peace. Get problem-solving help today. Thank you so much for coming to Hospital Near Me Medical Career YouTube channel. I am creating videos for people who want to get into medical school and other professions so they can work and serve as doctors and other healthcare professionals. Today, let's prepare you for the MCAT test so that you can get into medical school and become a doctor. Come and take my easy MCAT exam. If you are getting value from this channel, all I ask is that you let my ads play on the video, and if you could bless my PayPal, Cash App, Zelle, Buy Me a Coffee, or Patreon, it would be greatly appreciated. I am saving up for a new home as my old one was damaged by Hurricane Sandy. Thank you so much for your kindness. Please remember to like, subscribe, and comment below on my video. Now let's get started. Question 1. White blood cells. These defenders of our immune system play a critical role in protecting our bodies from pathogens and foreign invaders. White blood cells, also known as leukocytes, play a crucial role in the immune system, defending the body against infections and foreign invaders. There are several types of white blood cells, each with specific functions and characteristics. Understanding these cells is essential for the Medical College Admission Test, MCAT, as it assesses knowledge in the biological and biochemical foundations of living systems. Here's a brief overview of key white blood cell types. Neutrophils. Function. Neutrophils are the most abundant white blood cells and are primarily involved in phagocytosis, engulfing and destroying bacteria and other pathogens. Characteristics. They have a multi-lobed nucleus and are often the first responders during infection. Lymphocytes. Function. Lymphocytes are crucial for immune responses. There are two main types, T lymphocytes, T cells, and B lymphocytes, B cells. Characteristics. T lymphocytes, T cells. These cells directly attack infected cells and coordinate immune responses. B lymphocytes, B cells. B cells produce antibodies that target and neutralize pathogens. Monocytes. Function. Monocytes are involved in phagocytosis and serve as precursors to macrophages. Characteristics. They have a kidney-shaped nucleus and play a role in tissue repair and inflammation. Eosinophils. Function. Eosinophils are important in defending against parasitic infections and modulating allergic reactions. Characteristics. They have bilobed nuclei and contain granules that release enzymes. Basophils. Function. Basophils release histamine and other chemicals involved in inflammatory responses. Characteristics. They have large, dark staining granules and are involved in allergic reactions. Understanding the normal ranges of these white blood cells is crucial for assessing immune system health. Abnormalities in white blood cell counts may indicate various medical conditions, making this knowledge relevant for medical professionals. Remember, the MCAT may also test your understanding of cytokines, chemotaxis, and the overall immune response. So a comprehensive understanding of white blood cell functions is essential for success on the exam. Cytokines are small proteins that play a crucial role in cell signaling, regulating immune responses, inflammation, and hematopoiesis. Understanding cytokines is essential for individuals preparing for the Medical College Admission Test, MCAT, as they are integral to various physiological and pathological processes.
Cytokines are signaling molecules that mediate communication between cells of the immune system. Cytokines are signaling molecules that mediate communication between cells of the immune system. Cytokines are signaling molecules that mediate communication between cells of the immune system. Cytokines are signaling molecules that mediate communication between cells of the immune system. They can be classified into different categories based on their function, including interleukins, IL, interferons, IFN, tumor necrosis factors, TNF, chemokines, and growth factors. Cytokines are produced by various immune cells, such as T cells, B cells, macrophages, and dendritic cells. Different cytokines are synthesized in response to specific stimuli, leading to a cascade of signaling events. Cytokines play a pivotal role in regulating immune responses. For example, IL-2 is crucial for the activation and proliferation of T cells, while IFNs contribute to antiviral defense. TNF is involved in inflammation and the immune response against tumors. Certain cytokines, such as IL-1, IL-6, and TNF, are key mediators of inflammation. They induce fever, activate immune cells, and promote the acute phase response. Chronic dysregulation of cytokines can contribute to inflammatory diseases. Dysregulation of cytokines is implicated in various diseases. Excessive cytokine production can lead to autoimmune disorders, whereas insufficient cytokine responses may result in immunodeficiency. Targeting cytokines is a therapeutic approach in conditions like rheumatoid arthritis. Chemokines are a subset of cytokines responsible for directing cell migration. They play a crucial role in immune cell trafficking to sites of infection or inflammation. Understanding chemokine receptors is essential for comprehending immune cell movement. Growth factors, including cytokines like granulocyte colony-stimulating factor, GCSF, and erythropoietin, regulate the production of blood cells in a process known as hematopoiesis. This is crucial for maintaining a functional immune system. The knowledge of cytokines is applied in the development of therapeutic interventions. Biologics, such as monoclonal antibodies targeting specific cytokines, are used in the treatment of autoimmune diseases. Understanding the roles and interactions of cytokines is fundamental for medical professionals, especially those pursuing careers in immunology, infectious diseases, and rheumatology. The MCAT may test candidates on their comprehension of cytokines in the context of immune system function and pathology. Chemotaxis is a biological phenomenon that refers to the directed movement of cells or organisms in response to chemical gradients in their environment. This process is crucial for various physiological functions, including immune responses, wound healing, and the navigation of cells during embryonic development. Understanding chemotaxis is essential for students preparing for the Medical College Admission Test, MCAT, as it relates to several biological and cellular processes. Chemotaxis involves the movement of cells in response to chemical signals. It plays a vital role in the immune system, allowing white blood cells to migrate towards sites of infection or inflammation. Cells sense chemical gradients through receptors on their surface. Receptor binding triggers intracellular signaling pathways that lead to cytoskeletal rearrangements and changes in cell motility. Cells are capable of detecting even subtle differences in chemical concentrations. Gradient sensing mechanisms include biased random walks and spatial sensing, allowing cells to move accurately towards the source of the chemical signal. Various cell types exhibit chemotaxis, including neutrophils, macrophages, and certain types of cancer cells. In developmental biology, chemotaxis guides cells to specific locations during embryonic development. Chemical signals that induce chemotaxis are often referred to as chemotactic factors. Examples include cytokines, chemokines, and other signaling molecules. Dysregulation of chemotaxis can lead to pathological conditions such as chronic inflammation or autoimmune diseases. Cancer metastasis is often facilitated by chemotaxis, as malignant cells migrate towards blood vessels and invade distant tissues. Researchers use various experimental techniques to study chemotaxis, including microfluidic devices, in vitro assays, and advanced imaging methods. Understanding chemotaxis at the molecular and cellular levels contributes to the development of targeted therapies and pharmacological interventions. In summary, a comprehensive understanding of chemotaxis is crucial for aspiring medical professionals as it connects various aspects of cell biology, immunology, and developmental biology. Being familiar with the molecular mechanisms and clinical implications of chemotaxis will serve students well in the biological sciences section of the MCAT. The immune response is a complex and highly coordinated system that plays a crucial role in defending the body against pathogens such as bacteria, viruses, and fungi. This intricate defense mechanism involves the collaboration of various cells, proteins, and tissues to recognize and eliminate foreign invaders. There are two main components of the immune system: the innate immune system and the adaptive immune system. Innate immune system, physical barriers. The first line of defense includes physical barriers like the skin and mucous membranes that prevent pathogens from entering the body. Phagocytic cells, cells such as neutrophils and macrophages, engulf and digest pathogens. Inflammatory response. Inflammation is triggered to recruit immune cells to the site of infection, enhance blood flow, and eliminate pathogens. Adaptive immune system. Antigen presentation. Antigens from pathogens are presented to T cells by antigen-presenting cells, APCs, such as dendritic cells. T-cell activation. Helper T cells recognize antigens and stimulate B-cell and cytotoxic T cells. B-cell response. B cells differentiate into plasma cells, producing antibodies that specifically target and neutralize pathogens. Cell-mediated immunity. Cytotoxic T cells directly destroy infected cells. Memory response. Memory B and T cells. After an infection is cleared, memory B and T cells persist, providing rapid and targeted responses upon re-exposure to the same pathogen. Vaccination. Vaccines stimulate the immune system to produce memory cells without causing the disease, conferring immunity. Cytokines and signaling. Cell communication. Various signaling molecules, such as cytokines, coordinate immune responses by regulating cell activity and communication. Chemotaxis. Immune cells are guided to the site of infection by chemical signals. Immune disorders. Autoimmune diseases. The immune system mistakenly targets the body's own cells, leading to conditions like rheumatoid arthritis and lupus. Immunodeficiency. Conditions where the immune system is compromised, making individuals more susceptible to infections. Understanding the intricacies of the immune response is crucial for medical professionals, as it forms the basis for devising strategies to combat infectious diseases and developing vaccines. This knowledge is particularly relevant for aspiring healthcare practitioners, including those preparing for the MCAT, as it assesses their understanding of biological systems and their applications in medical contexts. The digestive system is a complex physiological system responsible for breaking down ingested food into nutrients that can be absorbed and utilized by the body. This system consists of various organs, each playing a crucial role in the process of digestion. Chemical digestion, a key component of this process, involves the breakdown of complex molecules into simpler ones through the action of enzymes. The digestive system begins with the oral cavity, where mechanical digestion occurs through chewing and saliva is released. Saliva contains the enzyme amylase, which initiates the breakdown of carbohydrates into simpler sugars like maltose. The partially digested food, known as bolus, then moves through the esophagus via peristalsis, a series of coordinated muscle contractions. In the stomach, gastric juices containing hydrochloric acid and pepsin are released. Pepsin is an enzyme that breaks down proteins into smaller peptides. The acidic environment in the stomach also helps to denature proteins, making them more susceptible to enzymatic action. After the stomach, the partially digested food enters the small intestine. Here, the pancreas releases pancreatic juices containing enzymes such as amylase, lipase, and protease. Amylase continues the breakdown of carbohydrates, lipase acts on fats, and protease further digests proteins into amino acids.
The small intestine also receives bile from the liver and gallbladder. Bile aids in the emulsification of fats, breaking them down into smaller droplets, which increases the surface area for enzymatic action. The final stage of digestion occurs in the small intestine, where the majority of nutrient absorption takes place. Nutrients including glucose, amino acids, and fatty acids are absorbed through the intestinal walls and transported to the bloodstream. The remaining undigested material moves into the large intestine, where water and electrolytes are absorbed, and the remaining material is formed into feces for elimination. In summary, the digestive system undergoes a series of intricate processes involving mechanical and chemical digestion to convert ingested food into absorbable nutrients. Understanding these processes is essential for success on the MCAT, as questions related to the digestive system frequently appear in the biology and biochemistry sections of the exam. Question 3. Survival and evolution. How has evolution shaped our ability to adapt and survive in changing environments? Survival traits are essential characteristics that contribute to the ability of a species to thrive and reproduce in its environment. These traits can be broadly categorized into physical and behavioral adaptations. Understanding these adaptations is crucial for students preparing for the MCAT, as they may encounter questions related to evolutionary biology and ecology. Here's a college-level overview of physical and behavioral survival traits. Physical survival traits. Structural adaptations. Camouflage. Species may develop coloration or patterns that blend with their surroundings, providing a defensive mechanism against predators. Mimicry. Some species evolve to resemble other organisms or objects, gaining protection by appearing harmful or inconspicuous. Physiological adaptations. Temperature regulation. Species adapt to specific temperature ranges through mechanisms like thermoregulation, ensuring optimal metabolic function. Desiccation resistance. Species in arid environments may develop adaptations to minimize water loss, such as water-retaining structures or behavioral strategies. Morphological features. Specialized appendages. Adaptations like claws, beaks, or fins enhance the ability to obtain food, defend against predators, or navigate specific environments. Protective coverings. Shells, spines, or scales provide protection against external threats. Behavioral survival traits. Feeding and foraging strategies. Social hunting. Cooperative hunting enhances the efficiency of obtaining prey, as seen in species like wolves or lions. Tool use. Some species exhibit tool use to extract or manipulate food resources, showcasing advanced cognitive abilities. Reproductive behaviors. Mating rituals. Elaborate courtship displays and rituals can attract mates and ensure successful reproduction. Parental care. Investment in offspring through parental care, including feeding, protection, and teaching, increases the chances of offspring survival. Communication and cooperation. Communication systems. Species with sophisticated communication systems, such as complex vocalizations or chemical signaling, can coordinate group activities or warn of potential threats. Cooperative defense. Group cooperation for defense against predators, as observed in meerkats or bees, enhances the survival chances of individuals. Adaptation to environmental changes. Migration. Seasonal migration allows species to follow resources and avoid harsh environmental conditions. Hibernation and dormancy. Some species enter states of reduced metabolic activity to survive unfavorable conditions. Understanding these physical and behavioral survival traits provides insights into how organisms have evolved to thrive in diverse ecological niches. This knowledge is relevant to the MCAT, especially in sections related to biology and evolution. Survival traits play a crucial role in the success and adaptation of a species. These traits can be broadly categorized into physical and behavioral characteristics. Understanding these traits is essential for individuals preparing for the Medical College Admission Test, MCAT. Here's a college-level overview of physical and behavioral survival traits. Physical survival traits. 1. Adaptations for thermoregulation. Species that can regulate their body temperature efficiently have a survival advantage. For instance, endothermic organisms, like mammals and birds, have a constant internal temperature, allowing them to thrive in a wider range of environments. Number 2. Protective coverings and structures. Physical features such as fur, scales, or exoskeletons provide protection against environmental elements, predators, and injuries. These adaptations contribute to the overall survival of the species. Number 3. Efficient locomotion. The ability to move effectively within the environment is a critical survival trait. Limb structure, wings, fins, or specialized appendages are examples of adaptations that enhance mobility and increase the chances of survival. 4. Respiratory adaptations. Species have evolved various respiratory systems to extract oxygen from their environment. Understanding the diversity of respiratory adaptations, such as gills, lungs, or spiracles, is important for comprehending how organisms survive in different habitats. Number 5. Metabolic efficiency. Efficient energy utilization is vital for survival. Species with adaptations that enhance metabolic efficiency, such as specialized digestive systems, are better equipped to extract nutrients from their diet, increasing their chances of survival. Behavioral survival traits. 1. Social structures. Many species exhibit social behaviors that contribute to their survival. From pack hunting to cooperative parenting, social structures enhance protection, resource acquisition, and overall survival rates. 2. Communication systems. Effective communication is crucial for coordinating activities within a group. Species with sophisticated communication systems, including vocalizations, visual cues, or chemical signals, often have a survival advantage. 3. Foraging and feeding strategies. Behavioral adaptations related to finding and acquiring food are essential. Examples include migration patterns, hunting strategies, and food storage behaviors, all of which contribute to the species' survival. 4. Territorial behavior. Defending a territory can provide access to resources and reduce competition. Understanding how species establish and maintain territories is important in studying their survival strategies. 5. Reproductive strategies. Species employ various reproductive strategies, such as mating rituals, parental care, or reproductive timing, to ensure the survival of their offspring. These behaviors are integral to the continuation of the species. Understanding the interplay between physical and behavioral survival traits is fundamental for comprehending the diversity of life on Earth. The knowledge of these adaptations is not only relevant for the MCAT, but also for a broader understanding of biology and ecology. Question 4. Signal transduction. Discover how cells communicate and coordinate responses to internal and external stimuli. Receptor-mediated signal transduction is a crucial cellular process that plays a central role in communication between cells. This intricate mechanism involves the binding of signaling molecules, often hormones or growth factors, to specific receptors on the cell membrane. The information carried by these signaling molecules is then transduced into the cell, leading to various cellular responses. 1. Receptor types. G-protein-coupled receptors, GPCRs. GPCRs are integral membrane proteins that span the cell membrane 7 times. Upon ligand binding, these receptors activate G proteins, initiating a cascade of intracellular events. Receptor tyrosine kinases, RTKs. RTKs are transmembrane proteins with intrinsic kinase activity. Ligand binding induces receptor dimerization and autophosphorylation, triggering downstream signaling cascades. Ion channel receptors. Ligand binding to these receptors alters their conformation, leading to the opening or closing of ion channels, thereby influencing cellular ion concentrations. Nuclear receptors. These receptors are located in the cell nucleus and regulate gene expression in response to ligand binding. Number 2. Ligand binding and receptor activation. Specificity. Each receptor has a high degree of specificity for its ligand, ensuring that only the appropriate signaling molecule triggers a response. Affinity and saturation. Receptors exhibit varying affinities for ligands, and the response is often dose-dependent. Saturation occurs when all available receptors are occupied. 3. Transduction pathways. Second messengers, small intracellular molecules such as CAMP, CGMP, IP3, and calcium ions often act as second messengers, relaying the signal from the cell membrane to the nucleus. Kinases and phosphatases. Phosphorylation and dephosphorylation of proteins by kinases and phosphatases, respectively, play a crucial role in signal transduction cascades.
Cellular responses, gene expression, activation of transcription factors, and alteration of gene expression patterns influence cellular functions and responses. Enzyme activation and inhibition. Signal transduction pathways often involve the activation or inhibition of enzymes, influencing metabolic processes within the cell. Termination of signal, desensitization. Receptor desensitization mechanisms, such as phosphorylation and internalization, prevent prolonged cellular responses to continuous ligand exposure. Negative feedback. Some signaling pathways include negative feedback loops to regulate the intensity and duration of the cellular response. Understanding receptor-mediated signal transduction is essential not only for cell biology, but also for comprehending the physiological processes and potential targets for therapeutic interventions. In the context of the MCAT test, a comprehensive knowledge of these concepts is crucial for success in the biology and biochemistry sections. The cellular organelle responsible for digestion and recycling materials within a cell is the lysosome. Lysosomes are membrane-bound organelles containing various hydrolytic enzymes that facilitate the breakdown of macromolecules such as proteins, lipids, nucleic acids, and carbohydrates. This process, known as hydrolysis, involves the enzymatic cleavage of bonds within these macromolecules, ultimately leading to their degradation into smaller, more manageable components. Key functions of lysosomes include intracellular digestion, waste disposal, and recycling of cellular materials. Lysosomes play a crucial role in maintaining cellular homeostasis by removing damaged organelles and cellular debris through a process called autophagy. During autophagy, lysosomes fuse with autophagosomes, which are vesicles containing cellular components targeted for degradation. The hydrolytic enzymes within lysosomes then break down the contents of autophagosomes, allowing the cell to recycle the building blocks for the synthesis of new molecules. On the MCAT, Medical College Admission Test, understanding the role of lysosomes in cellular digestion, waste disposal, and recycling is essential for questions related to cell biology, biochemistry, and physiology. Additionally, knowledge of lysosomal storage disorders, where malfunctioning lysosomes lead to the accumulation of undigested materials within cells, may also be relevant to MCAT exam content. These disorders can have significant implications for cellular function and overall health. Question 5. Role of lysosomes in cellular homeostasis. Learn about the essential role of lysosomes in maintaining cellular balance and recycling waste. Lysosomes and autophagy are crucial components of cellular biology, and understanding their functions is essential for any comprehensive study of cell biology. Here is college-level information about lysosomes and autophagy that may be relevant for the MCAT test. Lysosomes. Definition. Lysosomes are membrane-bound organelles found in the cytoplasm of eukaryotic cells. They are often referred to as the digestive system of the cell due to their role in breaking down and recycling cellular waste. Structure. Lysosomes are characterized by their single membrane and contain a variety of hydrolytic enzymes. These enzymes are responsible for catalyzing the hydrolysis of macromolecules, such as proteins, lipids, nucleic acids, and carbohydrates. Function. Intracellular digestion. Lysosomes play a key role in intracellular digestion by fusing with endocytic vesicles that contain material ingested by the cell through processes like phagocytosis or pinocytosis. Autophagy. Lysosomes are involved in autophagy, a process where cellular components, such as damaged organelles or misfolded proteins, are engulfed by autophagosomes and delivered to lysosomes for degradation. Waste recycling. Lysosomes break down the digested material into smaller molecules, which can be recycled by the cell for the synthesis of new cellular components. Programmed cell death. Apoptosis. Lysosomes are implicated in apoptosis, or programmed cell death, by releasing enzymes that lead to the degradation of cellular components, ultimately resulting in the controlled death of the cell. Autophagy. Definition. Autophagy is a cellular process that involves the degradation and recycling of cellular components through the formation of double-membrane structures called autophagosomes. Steps in autophagy. Initiation. Autophagy is initiated in response to cellular stress, nutrient deprivation, or the need for the removal of damaged organelles. Key regulatory proteins, such as mTOR, are involved in autophagy initiation. Autophagosome formation. During autophagy, a membrane sac called the phagophore elongates and engulfs cellular components destined for degradation, forming an autophagosome. Lysosomal fusion. The autophagosome fuses with a lysosome, forming an autolysosome. This fusion allows the lysosomal enzymes to degrade the sequestered material. Degradation. Within the autolysosome, the engulfed material is broken down into its constituent components by lysosomal enzymes, releasing them for cellular recycling. Physiological significance. Autophagy is essential for maintaining cellular homeostasis by eliminating damaged organelles, clearing misfolded proteins, and providing a source of nutrients during periods of nutrient scarcity. Understanding the intricate relationship between lysosomes and autophagy is critical for comprehending cellular maintenance and survival mechanisms. The dysregulation of these processes has been implicated in various diseases, making them important targets for therapeutic interventions. Lysosomal storage disorders, LSDs, encompass a group of rare genetic diseases characterized by the accumulation of undegraded substances within lysosomes, leading to cellular dysfunction and tissue damage. These disorders are typically caused by deficiencies in specific lysosomal enzymes, impeding the normal breakdown of macromolecules. Overview of lysosomes. Lysosomes are membrane-bound organelles within cells responsible for the degradation and recycling of cellular waste. They contain various hydrolytic enzymes that break down macromolecules such as lipids, proteins, and carbohydrates. Genetic basis of LSDs. LSDs are primarily genetic disorders resulting from mutations in genes that encode lysosomal enzymes or proteins involved in lysosomal function. Inheritance patterns can vary, with autosomal recessive being common. Accumulation of substrates. Due to enzyme deficiencies, substrates that would normally be broken down by lysosomal enzymes accumulate within the lysosomes. Examples of accumulated substances include glycosaminoglycans, sphingolipids, and glycogen. Clinical manifestations. LSDs can affect various organ systems, leading to a wide range of clinical symptoms. Common manifestations include neurodegeneration, organomegaly, skeletal abnormalities, and impaired motor and cognitive functions. Examples of LSDs: Gaucher's disease, caused by a deficiency of glucocerebrosidase, leading to the accumulation of glucocerebroside. Niemann-Pick disease, results from defects in sphingomyelinase, causing sphingomyelin accumulation. Tay-Sachs disease arises from a deficiency of hexosaminidase A, leading to GM2 ganglioside accumulation. Diagnosis and treatment. Diagnosis often involves genetic testing, enzyme assays, and imaging studies. Enzyme replacement therapy and substrate reduction therapy are among the treatment approaches, though they may not be curative. Research and future perspectives. Ongoing research focuses on developing gene therapies, enzyme replacement strategies, and small molecule approaches to address the underlying genetic defects. Understanding the molecular mechanisms of LSDs is crucial for developing targeted and effective therapeutic interventions. Understanding lysosomal storage disorders is important not only for medical professionals, but also for researchers aiming to develop innovative treatments for these rare and debilitating conditions. Lysosomal waste disposal is a crucial cellular process that plays a fundamental role in maintaining cellular homeostasis and preventing the accumulation of cellular waste products. Lysosomes are membrane-bound organelles containing hydrolytic enzymes that are responsible for breaking down various macromolecules, including proteins, lipids, nucleic acids, and carbohydrates. This process is essential for recycling cellular components and eliminating unwanted or damaged materials. Key points related to lysosomal waste disposal that might appear in a college-level discussion or on the MCAT test include: lysosome structure and function. Lysosomes are membrane-bound vesicles containing digestive enzymes such as proteases, lipases, nucleases, and glycosidases. These enzymes function optimally in the acidic environment of the lysosome, ensuring efficient degradation of macromolecules.
Autophagy is a cellular process through which cells can recycle their own components. It involves the sequestration of damaged organelles or cellular structures within double membrane vesicles called autophagosomes. Autophagosomes then fuse with lysosomes, allowing the contents to be degraded and recycled. The diverse array of enzymes present in lysosomes enables the breakdown of different types of biomolecules. For example, proteases break down proteins, lipases break down lipids, and glycosidases break down carbohydrates. Lysosomal waste disposal is critical for maintaining cellular homeostasis by preventing the accumulation of damaged or obsolete cellular components. Dysregulation of lysosomal function can lead to various diseases, including lysosomal storage disorders where cellular waste products accumulate due to defective enzymes. Endocytosis is a process where cells engulf extracellular material through vesicle formation. Once internalized, these vesicles fuse with lysosomes, leading to the degradation of the engulfed material. This mechanism is crucial for the clearance of extracellular debris and pathogens. Defects in lysosomal waste disposal pathways can result in lysosomal storage diseases, which are a group of genetic disorders characterized by the abnormal accumulation of undegraded materials within lysosomes. Enzyme replacement therapy and gene therapy are emerging strategies for treating lysosomal storage disorders. Understanding the intricacies of lysosomal waste disposal is not only essential for a comprehensive grasp of cellular biology, but also for appreciating the implications of dysregulation in various pathological conditions. This knowledge is relevant for medical students preparing for the MCAT, as questions related to cellular processes, molecular biology, and genetic disorders often appear on the exam. Lysosomes play a crucial role in cellular homeostasis, contributing to the maintenance of a stable internal environment within cells. These membrane-bound organelles contain various hydrolytic enzymes responsible for breaking down macromolecules, cellular waste, and foreign materials through a process known as autophagy. Lysosomes are spherical vesicles with an acidic interior, maintained by the action of proton pumps in their membrane. The enzymes within lysosomes function optimally in this acidic environment. Key enzymes include proteases, lipases, nucleases, and glycosidases. Lysosomes play a crucial role in the turnover of cellular components. They digest worn-out organelles and recycle their components, contributing to the renewal and maintenance of cellular structures. Autophagy is a process by which cells break down and recycle their own components. Lysosomes fuse with autophagosomes, double membrane vesicles containing cellular material, to degrade and recycle their contents. This process is vital for removing damaged organelles and maintaining cellular health. Lysosomes also participate in extracellular digestion. They fuse with phagosomes, which engulf extracellular materials, facilitating the degradation of ingested substances. This process is crucial for immune responses and defense against pathogens. Lysosomes contribute to the cell's defense mechanisms by aiding in the degradation of engulfed bacteria and other pathogens. This process, known as phagocytosis, is fundamental for the immune system's response to foreign invaders. Lysosomes play a role in detoxifying cells by breaking down harmful substances, preventing their accumulation and potential damage to cellular structures. Genetic mutations affecting lysosomal enzymes can lead to lysosomal storage disorders, where substrates accumulate within lysosomes. Examples include Tay-Sachs disease and Gaucher's disease. The decline in lysosomal function has been linked to cellular aging. Accumulation of cellular waste due to impaired lysosomal activity may contribute to age-related diseases. In summary, lysosomes are central to cellular homeostasis by maintaining a balance between synthesis and degradation. Their involvement in autophagy, extracellular digestion, and cellular defense highlights their critical role in sustaining cellular health and functionality. Understanding lysosomal function is essential for comprehending cellular physiology and the implications of lysosomal dysfunction in various pathological conditions. Meiotic division is a specialized type of cell division that occurs during the formation of gametes, sperm and egg cells, for sexual reproduction. Unlike mitotic division, which results in the production of identical daughter cells, meiotic division introduces genetic diversity through several key processes. During meiosis, homologous chromosomes with similar genetic information, pair up during prophase 1. Crossing over, or genetic recombination, occurs when sections of chromatids from homologous chromosomes break and exchange places. This process results in the mixing of genetic material between homologous chromosomes, leading to the creation of unique combinations of alleles on each chromatid. During metaphase 1 of meiosis, homologous chromosome pairs align at the cell's equator. The orientation of each pair is random, leading to the independent assortment of homologous chromosomes. This random alignment results in the potential formation of a vast number of different combinations of chromosomes in the resulting gametes. Meiosis consists of two sequential divisions, meiosis 1 and meiosis 2. Meiosis 1 is a reductional division where the chromosome number is halved. Each resulting cell has only one set of chromosomes, ensuring that gametes have half the genetic material of the parent cell. This reduction in chromosome number contributes to genetic diversity among offspring. After meiosis, haploid gametes are produced, each with a unique combination of genetic material. During fertilization, when sperm and egg cells combine, the resulting diploid zygote inherits a combination of genetic material from both parents. The chance of fertilization is random, further contributing to the diversity of genetic combinations in the offspring. Overall, meiotic division introduces genetic diversity through the processes of crossing over independent assortment, reductional division, and random fertilization. This diversity plays a crucial role in the variation observed among individuals within a population, contributing to the adaptability and evolution of species. Understanding these mechanisms is essential for a comprehensive grasp of reproductive biology and genetics, topics that are relevant for the MCAT exam. Arteries and valves are essential components of the circulatory system, playing crucial roles in maintaining blood flow and ensuring proper functioning of the cardiovascular system. Understanding these structures is fundamental for success on the MCAT, Medical College Admission Test. Here is a college-level overview of arteries and valves. Arteries are blood vessels that carry oxygenated blood away from the heart to various tissues and organs throughout the body. They are classified into three main types based on their size and function. Large vessels near the heart, such as the aorta, contain elastic fibers in their walls, allowing them to stretch and recoil with each heartbeat. Function to maintain continuous blood flow during diastole, relaxation phase of the heart. Medium-sized arteries that distribute blood to specific organs and body regions have a greater amount of smooth muscle in their walls, allowing for more precise control of blood flow. Small arteries that regulate blood flow into capillaries play a key role in controlling blood pressure and distributing blood based on the body's needs. Valves in the cardiovascular system prevent the backflow of blood and ensure unidirectional flow through the heart and major vessels. There are two primary types of valves, located between the atria and ventricles in the heart. The tricuspid valve is on the right side, while the bicuspid mitral valve is on the left side. Prevent backflow of blood from the ventricles to the atria during ventricular contraction.
Semilunar valves, positioned at the exits of the heart's ventricles. The pulmonary valve guards the entrance to the pulmonary artery from the right ventricle, while the aortic valve prevents backflow into the left ventricle from the aorta. Ensure that blood flows in one direction from the heart to the arteries. Understanding the anatomy and function of arteries and valves is crucial for medical students preparing for the MCAT. It provides a foundation for comprehending the complexities of the cardiovascular system and its role in maintaining homeostasis within the human body. Arteries, valves, and homeostasis in the cardiovascular system. Arteries. Arteries are blood vessels that carry oxygenated blood away from the heart to various parts of the body. They have thick, elastic walls composed of smooth muscle and connective tissue, allowing them to withstand the high pressure generated by the pumping action of the heart. The main types of arteries include elastic arteries, distributing arteries, and arterioles. Valves. While valves are more commonly associated with the heart, preventing the backflow of blood, there are no valves within arteries. Instead, valves are present in the heart and veins. In the heart, there are four valves: the tricuspid valve, the pulmonary valve, the mitral valve, and the aortic valve. These valves ensure the unidirectional flow of blood through the heart chambers, facilitating efficient circulation. Homeostasis in the cardiovascular system. Homeostasis refers to the body's ability to maintain a stable internal environment despite external fluctuations. In the cardiovascular system, several mechanisms contribute to homeostasis. Blood pressure regulation. The body regulates blood pressure to ensure an adequate supply of oxygen and nutrients to tissues. Baroreceptors, located in the walls of certain arteries, sense changes in blood pressure and signal the nervous system to adjust heart rate and blood vessel diameter accordingly. Oxygen and carbon dioxide balance. Homeostasis is also maintained by regulating the levels of oxygen and carbon dioxide in the blood. Chemoreceptors monitor these levels and trigger adjustments in respiratory rate to maintain the proper balance. Fluid and electrolyte balance. The cardiovascular system plays a crucial role in maintaining fluid and electrolyte balance. Blood vessels help regulate the distribution of fluids between the bloodstream and surrounding tissues, ensuring proper hydration. Temperature regulation. Blood vessels contribute to temperature regulation by adjusting blood flow to the skin. Vasodilation increases blood flow to dissipate heat, while vasoconstriction conserves heat by reducing blood flow. Understanding the intricate relationship between arteries, valves, and the mechanisms that maintain homeostasis in the cardiovascular system is essential for success on the MCAT, particularly in the biological and biochemical foundations of living systems section. Question 8. Pulmonary artery. Learn about the unique function of the pulmonary artery in the circulatory system. The pulmonary artery plays a crucial role in the circulatory system, specifically in the transportation of deoxygenated blood from the heart to the lungs for oxygenation. Understanding the pressure dynamics in the pulmonary artery is essential for grasping key physiological concepts tested on the MCAT. Normal pulmonary arterial pressure. The pressure in the pulmonary artery is lower compared to the systemic arteries. While systemic arterial pressure is typically measured in millimeters of mercury, mmHg, pulmonary arterial pressure is often expressed in a range from 8 to 20 mmHg for systolic pressure and 4 to 12 mmHg for diastolic pressure. This lower pressure is reflective of the fact that the pulmonary circulation is a low-pressure system designed for the efficient exchange of gases in the lungs. Pulmonary hypertension. Pulmonary hypertension is a condition characterized by elevated pressure in the pulmonary arteries. This can be caused by various factors, such as chronic lung diseases, heart failure, or disorders affecting the pulmonary vasculature. On the MCAT, understanding the consequences of pulmonary hypertension is crucial. It can lead to increased workload on the right ventricle of the heart, potentially resulting in right heart failure. Pressure gradients. The pressure in the pulmonary artery is influenced by pressure gradients during different phases of the cardiac cycle. During systole, when the right ventricle contracts, pressure increases in the pulmonary artery. In diastole, when the right ventricle relaxes, the pressure decreases. The pressure changes in a coordinated manner with the systemic circulation to ensure efficient blood flow through the pulmonary circulation and the subsequent oxygenation of blood in the lungs. Pulmonary arterial compliance. Compliance refers to the ability of the pulmonary arteries to expand and accommodate changes in blood volume without a significant increase in pressure. The compliance of the pulmonary arteries contributes to the efficient flow of blood and prevents excessive pressure buildup. Regulation of pulmonary arterial pressure. The autonomic nervous system, particularly the sympathetic and parasympathetic branches, plays a role in regulating pulmonary arterial pressure. Additionally, local factors such as oxygen levels in the lungs and the release of vasoactive substances influence pulmonary vascular resistance, and consequently, understanding these concepts about pulmonary arterial pressure is crucial for answering MCAT questions related to cardiovascular physiology and pathology. It's essential to integrate this knowledge with broader topics such as gas exchange, cardiac function, and hemodynamics to excel in the examination. The pulmonary artery is a vital blood vessel that plays a crucial role in the cardiovascular system. It is responsible for carrying deoxygenated blood from the right ventricle of the heart to the lungs for oxygenation. Understanding the anatomy and function of the pulmonary artery is essential for a comprehensive grasp of cardiovascular physiology and pathology, topics frequently tested on the MCAT. Anatomy of the pulmonary artery. The pulmonary artery is a large vessel originating from the right ventricle of the heart. It is divided into two main branches, the right and left pulmonary arteries, which further branch into smaller vessels within the lungs. These smaller vessels ultimately reach the pulmonary capillaries where gas exchange occurs. Function in cardiovascular physiology. The primary function of the pulmonary artery is to transport deoxygenated blood to the lungs, where it undergoes oxygenation. This oxygenation is crucial for the cellular respiration processes occurring in tissues throughout the body. The right ventricle pumps blood into the pulmonary artery, initiating the pulmonary circulation. During the pulmonary circulation, carbon dioxide is released from the blood into the lungs, and oxygen is absorbed from the air into the blood. This oxygenated blood is then returned to the left atrium via the pulmonary veins, completing the pulmonary circuit and setting the stage for systemic circulation. Pathology and implications for the MCAT. Understanding the pathology of the pulmonary artery is essential for tackling MCAT questions related to cardiovascular diseases. Pathological conditions associated with the pulmonary artery include pulmonary hypertension. This condition involves increased pressure within the pulmonary arteries, which can lead to strain on the right ventricle and potential heart failure. Causes include chronic lung diseases and certain cardiac conditions. Pulmonary embolism. A pulmonary embolism occurs when a blood clot or other material blocks one or more pulmonary arteries. This can lead to decreased blood flow to the lungs and impaired oxygenation. Congenital heart defects. Some congenital conditions involve malformations of the pulmonary artery, affecting blood flow and oxygenation. Knowledge of these defects is crucial for understanding cardiovascular abnormalities.
Physiological processes is often assessed. Question 10. The reflex arc. Discover the neural pathway that allows for rapid responses to stimuli through reflex actions. The reflex arc is a neural pathway that mediates a reflex action. It is an essential component of the nervous system and plays a crucial role in the body's ability to respond rapidly to stimuli. Understanding the reflex arc is pertinent to various medical and biological disciplines, making it a potential topic for the MCAT test. The reflex arc typically involves the following key components. Receptor. The process begins with a receptor, which is a specialized sensory nerve ending that detects a stimulus. This stimulus could be anything from a touch or pressure to a change in temperature. Sensory neuron. Once the receptor detects the stimulus, it sends signals through a sensory neuron,
also known as an afferent neuron, towards the central nervous system, CNS. The sensory neuron carries the information from the receptor to the spinal cord or brain, depending on the nature of the reflex. Integration center. In the CNS, the sensory neuron synapses with interneurons, association neurons, in the spinal cord or brain. This region is referred to as the integration center. Here, the signal is processed and a decision is made regarding the appropriate response to the stimulus. Motor neuron. Following integration, the processed signal is transmitted through a motor neuron, efferent neuron, that exits the CNS. This neuron carries the response signal from the CNS to the effector organ, usually a muscle or gland. Effector organ. The motor neuron synapses with the effector organ, which executes the response. If the reflex is a muscle contraction, the effector organ would be a muscle. If it involves secretion, the effector organ could be a gland. Response. The effector organ carries out the response to the stimulus, leading to a quick and involuntary reaction. Importantly, this response occurs without conscious thought and is designed to protect the body from potential harm. One classic example of a reflex arc is the patellar reflex, commonly tested during neurological examinations. In this reflex, tapping the patellar tendon just below the kneecap stretches the quadriceps muscle, leading to a reflexive contraction of the muscle. Understanding the reflex arc is crucial in the context of medical examinations like the MCAT, as it demonstrates knowledge of neural pathways, sensory processing, and motor responses within the nervous system. It also provides insights into how the body efficiently and rapidly responds to various stimuli. The reflex arc is a neural pathway that mediates a reflex action. It is a rapid and involuntary response to a stimulus, often serving as a protective mechanism for the body. Understanding the components of a reflex arc is crucial for anyone studying neurobiology, and it may be relevant for the MCAT test. Here is a breakdown of the key components of a typical reflex arc. Receptor. The process begins with a sensory receptor that detects a stimulus. This can be anything from a touch to a painful sensation. Sensory receptors are typically located in the skin, muscles, or internal organs. Sensory neuron, afferent neuron. Once the receptor is stimulated, sensory neurons transmit the signal to the central nervous system, CNS. These neurons are known as afferent neurons, as they carry information from the periphery to the CNS. Integration center, spinal cord or brain. The incoming sensory information reaches an integration center, which can be the spinal cord or specific regions of the brain. Here, the signal is processed and a decision is made on how to respond. Motor neuron, efferent neuron. Following integration, motor neurons, efferent neurons, are activated. These neurons carry the response signal from the CNS to the effector organ, which is usually a muscle or gland.
Effector organ. The final component is the effector organ, which carries out the response to the stimulus. If the reflex is a muscle contraction, the effector organ is a muscle. If it involves a gland, the gland becomes the effector organ. Response. The response is the actual action that occurs in reaction to the original stimulus. For example, if you touch a hot surface, the reflex arc will cause your muscles to quickly pull your hand away, preventing further damage. It's important to note that reflex arcs are often simple and involve only a few neurons. This allows for quick responses without the need for conscious thought. Moreover, some reflex arcs involve the spinal cord exclusively, monosynaptic reflexes, while others may involve the brain, polysynaptic reflexes. Understanding the reflex arc and neural pathways is crucial for comprehending the functioning of the nervous system. Additionally, it provides insight into how the body can swiftly and automatically respond to potentially harmful stimuli, showcasing the efficiency and complexity of neural networks. The reflex arc is a neural pathway that mediates a rapid, involuntary response to a stimulus. This process involves sensory input, integration in the central nervous system, CNS, and motor output. Understanding the reflex arc is crucial for comprehending sensory processing, and it is a topic that might appear on the MCAT, Medical College Admission Test. Here's a college-level overview. Stimulus reception. The reflex arc begins with the reception of a stimulus by a sensory receptor. Sensory receptors are specialized cells or nerve endings that detect changes in the environment. For example, in the patellar reflex, the stimulus is a tap on the patellar tendon. Sensory neuron. The sensory neuron, also known as the afferent neuron, carries the sensory information from the receptor to the CNS. These neurons are typically pseudo-unipolar, with a single process extending from the cell body. Dorsal root ganglion. The sensory neuron enters the spinal cord through the dorsal root. Near the entry point, there is a swelling called the dorsal root ganglion, which contains the cell bodies of sensory neurons. Integration in the CNS. The sensory information is then transmitted to an interneuron, association neuron, in the spinal cord or brainstem. This is where integration occurs. The interneuron processes the information and decides on the appropriate response. Motor neuron. The motor neuron, or efferent neuron, carries the response generated by the integration to the effector organ or muscle. This neuron is often multipolar and extends from the CNS to the effector. Effector organ. The effector organ is the target tissue that carries out the response. In the case of the patellar reflex, the effector organ is the quadriceps muscle, which contracts in response to the tap on the patellar tendon. Response. The motor neuron stimulates the effector organ, resulting in a quick and involuntary response. In the patellar reflex, this leads to the contraction of the quadriceps muscle, causing the leg to kick. Monosynaptic versus polysynaptic reflexes. Some reflex arcs involve a single synapse between the sensory and motor neurons, monosynaptic reflexes, while others involve one or more interneurons, polysynaptic reflexes. The patellar reflex is an example of a monosynaptic reflex. Understanding the reflex arc is essential not only for grasping sensory processing, but also for appreciating the complexity of neural pathways involved in basic motor responses. This knowledge is foundational for medical students, particularly those pursuing a career in neurology or related fields. The reflex arc is a neural pathway that mediates a reflex action. It involves the rapid and involuntary response to a stimulus without conscious thought. Motor responses within the reflex arc are crucial components tested on the MCAT, Medical College Admission Test, as they demonstrate an understanding of neurophysiology. Here is a college-level overview of reflex arc motor responses. Stimulus reception. The reflex arc begins with the detection of a stimulus by a sensory receptor, such as a pain receptor, nociceptor, or stretch receptor, proprioceptor. The stimulus generates action potentials in the sensory neuron, which is typically a pseudo-unipolar neuron. Sensory neuron transmission. Action potentials travel along the sensory neuron towards the central nervous system, CNS. In the spinal reflex arc, this often involves the dorsal root ganglion. Integration in the CNS. The sensory neuron synapses with interneurons in the spinal cord or brainstem, allowing for integration of the sensory information. Interneurons play a crucial role in relaying signals and modulating the reflex response. Some reflex arcs involve a single synapse, monosynaptic, while others involve multiple synapses, polysynaptic. Motor neuron activation. Interneurons stimulate the motor neurons in the ventral horn of the spinal cord or the brainstem. The motor neurons are typically alpha motor neurons that innervate skeletal muscles. Effector organ response. Action potentials generated in the motor neurons travel down the efferent pathway towards the effector organ, usually a muscle. At the neuromuscular junction, acetylcholine is released, leading to muscle contraction. Muscle contraction. The motor response involves the contraction of the effector muscle, which is the desired outcome of the reflex. This rapid and involuntary response is critical for maintaining homeostasis, avoiding harm, or responding to external stimuli. Feedback mechanism. In many reflex arcs, there is a feedback mechanism that regulates the response. For example, a reflex involving muscle stretch may lead to contraction or relaxation to maintain proper muscle tone. Understanding the reflex arc and its motor responses is fundamental for medical students as it relates to both normal physiological processes and clinical examinations. Moreover, it provides insight into the intricate coordination of sensory and motor pathways in the nervous system. The reflex arc is a neural pathway that controls a reflex action. Reflexes are rapid, automatic responses to specific stimuli that help protect the body from potential harm. Understanding the reflex arc is crucial for students preparing for the MCAT, as it involves concepts related to neurobiology and the nervous system. The reflex arc consists of several key components. Receptor. The process begins with a sensory receptor that detects a stimulus. This can be anything from touching a hot surface to a sudden loud noise. Sensory neuron. The sensory neuron, also known as an afferent neuron, transmits the information from the receptor to the central nervous system, CNS. In the context of the MCAT, it's important to know that this neuron typically enters the spinal cord. Integration center. In the spinal cord or brain, the sensory neuron synapses with an interneuron. The interneuron processes the information and determines the appropriate response.
Motor neuron. The processed signal then travels through a motor neuron, also known as an efferent neuron. This neuron carries the response signal from the CNS back to the effector organ or muscle. Effector organ or muscle. The motor neuron stimulates the effector organ or muscle, leading to the actual response. This can be a muscle contraction or relaxation, depending on the nature of the reflex. Response. The final step is the observable response to the stimulus. This response is typically quick and involuntary, designed to protect the body from potential harm. Understanding the reflex arc is crucial for medical students as it provides insights into the organization and function of the nervous system. Additionally, the reflex arc illustrates the concept of a feedback loop that enables rapid responses without conscious thought. In the context of the MCAT, questions may focus on the sequence of events in a reflex arc, the role of specific neurons, or the integration center's function. It's essential for students to grasp these concepts to perform well in the biology and physiology sections of the exam. Question 1. Which type of white blood cell is primarily responsible for the production of antibodies? A. Neutrophils. B. Basophils. C. T lymphocytes, T cells. D. Monocytes. Question 2. Which of the following statements accurately describes the role of the pancreas in the digestive system? A. The pancreas produces bile, which aids in the emulsification of fats. B. The pancreas secretes insulin to regulate blood sugar levels. C. The pancreas synthesizes digestive enzymes that break down carbohydrates in the stomach. D. The pancreas stores and releases bile to assist in the digestion of proteins. Question 3. Which of the following physical and behavioral survival traits would likely provide a species with a significant advantage in its environment? A. Enhanced camouflage abilities. B. Rapid reproduction rate. C. Strict territorial behavior. D. Lack of social cooperation. Question 4. Which of the following describes the initial step in receptor-mediated signal transduction? A. Phosphorylation of the receptor. B. Ligand binding to the receptor. C. Internalization of the receptor-ligand complex. Activation of downstream signaling molecules. Question 5. Which of the following statements accurately describes the role of lysosomes in cellular homeostasis and their association with lysosomal storage disorders? A. Lysosomes are primarily involved in energy production, and lysosomal storage disorders result from excessive lysosomal activity. B. Lysosomes play a key role in cellular waste management, and lysosomal storage disorders are characterized by a deficiency in lysosomal enzymes. C. Lysosomes regulate cell division, and lysosomal waste disease is caused by an overabundance of lysosomal membranes. D. Lysosomes are responsible for protein synthesis, and lysosomal storage disorders arise from a surplus of lysosomal transporters. Question 6. Which of the following statements best explains the role of diversity in meiotic division as opposed to mitotic division? A. Meiotic division promotes genetic diversity by crossing over between homologous chromosomes. B. Mitotic division generates genetic diversity through the fusion of gametes. C. Meiotic division produces genetically identical daughter cells. D. Mitotic division involves the separation of homologous chromosomes. Question 7. Which of the following statements accurately describes the role of arteries, valves, and homeostasis in the circulatory system? A. Arteries contain valves that prevent the backflow of blood. B. Arteries carry deoxygenated blood away from the heart. C. Valves in arteries help regulate blood pressure. D. Homeostasis is primarily maintained by the contraction and relaxation of veins. Question 8. Which of the following statements accurately describes the pressure in the pulmonary artery? A. The pressure in the pulmonary artery is higher than the systemic arterial pressure. B. The pressure in the pulmonary artery is lower than the systemic venous pressure. C. The pressure in the pulmonary artery is higher during diastole. D. The pressure in the pulmonary artery is lower during inspiration. Question 9. In the lumen of the small intestine, sodium bicarbonate plays a crucial role in A. Lipid digestion, B. Carbohydrate absorption, C. pH regulation, D. Protein synthesis. Question 10. Which of the following accurately describes the sequence of events in a reflex arc? A sensory neuron receives stimuli, sends signal to effector, motor neuron processes information. B. Motor neuron receives stimuli, sends signal to effector, sensory neuron processes information. C. Sensory neuron receives stimuli, processes information, sends signal to effector. D. Motor neuron receives stimuli, processes information, sends signal to effector. Now let's give you the answers to the test questions. Question 1. Which type of white blood cell is primarily responsible for the production of antibodies? A. Neutrophils, B. Basophils, C. T lymphocytes, T cells, D. Monocytes. Correct answer C. T lymphocytes, T cells. Explanation. T lymphocytes, or T cells, play a central role in the immune system and are primarily responsible for the production of antibodies. T cells are involved in cell-mediated immunity and coordinate the immune response by activating and regulating other immune cells, including B cells. B cells, not mentioned in the options, are the ones that produce antibodies. T cells help in the activation of B cells, which then differentiate into plasma cells, producing antibodies. Incorrect options. A. Neutrophils. Neutrophils are phagocytic cells that are important for the initial defense against bacterial infections. They are not directly involved in antibody production. B. Basophils. Basophils release histamine and are involved in allergic reactions and inflammation. They are not responsible for antibody production. D. Monocytes. Monocytes are precursors to macrophages and dendritic cells. While they have a role in phagocytosis and immune response, they are not the primary cells responsible for antibody production. Question 2. Which of the following statements accurately describes the role of the pancreas in the digestive system? A. The pancreas produces bile, which aids in the emulsification of fats. B. The pancreas secretes insulin to regulate blood sugar levels. C. The pancreas synthesizes digestive enzymes that break down carbohydrates in the stomach. D. The pancreas stores and releases bile to assist in the digestion of proteins. Correct answer, B. The pancreas secretes insulin to regulate blood sugar levels. Explanation. A. This statement is incorrect. The pancreas does not produce bile. That function is attributed to the liver. B. This is the correct answer. The pancreas is responsible for secreting insulin, a hormone that regulates blood sugar levels. C. This statement is incorrect. While the pancreas does produce digestive enzymes, they primarily target proteins, not carbohydrates. D. This statement is incorrect. The storage and release of bile for protein digestion occur in the gallbladder, not the pancreas. Question 3. Which of the following physical and behavioral survival traits would likely provide a species with a significant advantage in its environment? A. Enhanced camouflage abilities, B. Rapid reproduction rate, C. Strict territorial behavior, D. Lack of social cooperation. Correct answer, A. Enhanced camouflage abilities. Explanation. Enhanced camouflage abilities offer a species a significant advantage in its environment by increasing its chances of avoiding predators or capturing prey. Camouflage helps the organism blend into its surroundings, making it less visible to predators or unsuspecting prey. This trait is particularly crucial for survival in diverse ecosystems, where visual detection plays a key role in predator-prey dynamics. Option B, rapid reproduction rate, may seem advantageous, but it can lead to overpopulation and resource depletion, potentially causing more harm than good in the long run. Rapid reproduction may not always be sustainable, especially if the environment cannot support a large population. Option C, strict territorial behavior, can be advantageous in certain situations, but it may also lead to conflicts with neighboring individuals or groups. Cooperation and adaptability are essential for long-term survival, and strict territorial behavior might limit a species' ability to respond effectively to changing environmental conditions. Option D, lack of social cooperation, is generally disadvantageous for species survival. Social cooperation often provides benefits such as shared resources, protection, and division of labor, contributing to the overall fitness of the group. Lack of social cooperation may result in decreased chances of survival in complex ecosystems where teamwork is essential. In summary, enhanced camouflage abilities, option A, stand out as the most advantageous trait among the given options, as it directly contributes to the species' ability to evade predators and secure prey while maintaining adaptability in its environment. Question 4. Which of the following best describes the initial step in receptor-mediated signal transduction? A. Phosphorylation of the receptor. B. Ligand binding to the receptor. C. Internalization of the receptor-ligand complex. D. Activation of downstream signaling molecules. Correct answer B. Ligand binding to the receptor. Explanation. Receptor-mediated signal transduction typically begins with the binding of a signaling molecule, ligand, to its specific receptor. This interaction is crucial as it initiates a cascade of events that transmit the extracellular signal into the cell. The binding of the ligand induces a conformational change in the receptor, triggering its activation.
Let's analyze the other options. A. Phosphorylation of the receptor. While receptor phosphorylation is a common event in signal transduction, it usually occurs after ligand binding. Ligand binding induces conformational changes that expose phosphorylation sites on the receptor, allowing for subsequent phosphorylation. C. Internalization of the receptor-ligand complex. Internalization typically occurs after ligand binding and activation of the receptor. It is a later step in the process and is involved in the regulation of receptor activity and signal termination. D. Activation of downstream signaling molecules. This is a downstream event that occurs after ligand binding and receptor activation. The initial step is the binding of the ligand to the receptor, which triggers the activation of downstream signaling molecules. In summary, the correct answer is B because ligand binding to the receptor is the initial and crucial step in receptor-mediated signal transduction. Question 5. Lysosomes. Which of the following statements accurately describes the role of lysosomes in cellular homeostasis and their association with lysosomal storage disorders? A. Lysosomes are primarily involved in energy production, and lysosomal storage disorders result from excessive lysosomal activity. B. Lysosomes play a key role in cellular waste management, and lysosomal storage disorders are characterized by a deficiency in lysosomal enzymes. C. Lysosomes regulate cell division, and lysosomal waste disease is caused by an overabundance of lysosomal membranes. D. Lysosomes are responsible for protein synthesis, and lysosomal storage disorders arise from a surplus of lysosomal transporters. Correct answer, B. Lysosomes play a key role in cellular waste management, and lysosomal storage disorders are characterized by a deficiency in lysosomal enzymes. Explanation. B. This is the correct answer. Lysosomes are cellular organelles involved in the digestion and recycling of cellular waste, including old or damaged organelles and macromolecules. Lysosomal storage disorders occur when there is a deficiency in lysosomal enzymes, leading to the accumulation of undigested substances within the lysosomes. A. This is incorrect. Lysosomes are not primarily involved in energy production. Lysosomal storage disorders result from a deficiency, not excessive activity. C. This is incorrect. Lysosomes do not regulate cell division. Lysosomal waste disease is not caused by an overabundance of lysosomal membranes. D. This is incorrect. Lysosomes are not responsible for protein synthesis. Lysosomal storage disorders result from a deficiency in lysosomal enzymes, not an excess of transporters. Question 6. Mitotic and meiotic division. Which of the following statements best explains the role of diversity in meiotic division as opposed to mitotic division? A. Meiotic division promotes genetic diversity by crossing over between homologous chromosomes. B. Mitotic division generates genetic diversity through the fusion of gametes. C. Meiotic division produces genetically identical daughter cells. D. Mitotic division involves the separation of homologous chromosomes. Correct answer, A. Meiotic division promotes genetic diversity by crossing over between homologous chromosomes. Explanation. A. This is the correct answer. Meiotic division is characterized by the process of crossing over, where genetic material is exchanged between homologous chromosomes. This results in the creation of genetically unique daughter cells, contributing to genetic diversity among offspring. B. This statement is incorrect. Genetic diversity is not generated through mitotic division, as mitosis leads to the formation of genetically identical daughter cells. C. This statement is incorrect. Meiotic division, unlike mitotic division, does not produce genetically identical daughter cells due to the occurrence of crossing over and the random assortment of chromosomes. D. This statement is incorrect. Mitotic division involves the separation of sister chromatids, not homologous chromosomes. Homologous chromosomes separate during meiotic division, contributing to genetic diversity. In summary, the correct answer is A, because meiotic division specifically introduces genetic diversity through the process of crossing over between homologous chromosomes, which is not a feature of mitotic division. Question 7. Arteries and valves. Which of the following statements accurately describes the role of arteries, valves, and homeostasis in the circulatory system? A. Arteries contain valves that prevent the backflow of blood. B. Arteries carry deoxygenated blood away from the heart. C. Valves and arteries help regulate blood pressure. D. Homeostasis is primarily maintained by the contraction and relaxation of veins. Correct answer C. Valves and arteries help regulate blood pressure. Explanation. A. This statement is incorrect. Arteries generally do not contain valves. Valves are more commonly found in veins to prevent the backflow of blood. This statement is incorrect. Arteries typically carry oxygenated blood away from the heart, not deoxygenated blood. C. The correct answer. Valves and arteries play a crucial role in regulating blood pressure by ensuring that blood flows in one direction. These valves prevent the backflow of blood and maintain proper circulation. D. This statement is incorrect. Homeostasis, the maintenance of stable internal conditions, is not primarily regulated by the contraction and relaxation of veins. Homeostasis involves the coordination of various physiological processes to maintain balance in the body. In summary, option C is the correct answer because valves and arteries contribute to the regulation of blood pressure, while the other options contain inaccuracies about the roles of arteries, valves, and homeostasis in the circulatory system.
Question 8. Which of the following statements accurately describes the pressure in the pulmonary artery? A. The pressure in the pulmonary artery is higher than the systemic arterial pressure. B. The pressure in the pulmonary artery is lower than the systemic venous pressure. C. The pressure in the pulmonary artery is higher during diastole. D. The pressure in the pulmonary artery is lower during inspiration. Correct answer, B. The pressure in the pulmonary artery is lower than the systemic venous pressure. Explanation. A. Incorrect. The pressure in the pulmonary artery is generally lower than the systemic arterial pressure. The pulmonary circulation has a lower resistance compared to the systemic circulation, resulting in lower pressures. B. Correct. The pressure in the pulmonary artery is indeed lower than the systemic venous pressure. This is because the blood is pumped from the right ventricle into the pulmonary artery, which then carries deoxygenated blood to the lungs. The pressure drop across the pulmonary circulation is necessary for efficient oxygenation of blood in the lungs. C. Incorrect. The pressure in the pulmonary artery is generally higher during systole, when the right ventricle contracts to pump blood into the pulmonary circulation. Diastole is the relaxation phase, and the pressure is lower during this phase. D. Incorrect. The pressure in the pulmonary artery is not significantly influenced by the respiratory cycle. It is more closely tied to the cardiac cycle, with variations occurring during systole and diastole. In summary, option B is the correct answer because it accurately reflects the relationship between the pressure in the pulmonary artery and the systemic venous pressure, taking into account the physiological dynamics of the pulmonary circulation. Question 9. In the lumen of the small intestine, sodium bicarbonate plays a crucial role in A. Lipid digestion, B. Carbohydrate absorption, C. pH regulation, D. Protein synthesis. Answer: C. pH regulation. Explanation: sodium bicarbonate, a weak base, is secreted by the pancreas into the small intestine to neutralize the acidic chyme arriving from the stomach. This alkaline environment, created by sodium bicarbonate, is essential for the optimal activity of digestive enzymes, especially those involved in the digestion of carbohydrates. The correct answer is thus pH regulation. Now let's analyze the other options. A. Lipid digestion. Sodium bicarbonate is not directly involved in lipid digestion. Lipid digestion primarily relies on bile salts, which emulsify fats for enzymatic breakdown. B. Carbohydrate absorption. While sodium bicarbonate helps create an optimal pH for carbohydrate digestion, it is not directly involved in carbohydrate absorption. Carbohydrate absorption mainly occurs in the small intestine through specialized transporters. D. Protein synthesis. Sodium bicarbonate is not involved in protein synthesis. Protein synthesis occurs at the cellular level, particularly in the ribosomes of cells, and is not influenced by the pH in the lumen of the small intestine. In summary, the correct answer is C. pH regulation, as sodium bicarbonate helps maintain an alkaline environment, facilitating the activity of digestive enzymes in the small intestine. Question 10. Which of the following accurately describes the sequence of events in a reflex arc? A. Sensory neuron receives stimuli, sends signal to effector, motor neuron processes information. B. Motor neuron receives stimuli, sends signal to effector, sensory neuron processes information. C. Sensory neuron receives stimuli, processes information, sends signal to effector. D. Motor neuron receives stimuli, processes information, sends signal to effector. Correct answer, C. Explanation. In a reflex arc, the sequence of events is as follows. Sensory neuron receives stimuli. Sensory receptors detect a stimulus, e.g. touch, heat, pain, and generate signals that are transmitted to the sensory neuron. Processes information. The sensory neuron processes the incoming signals in the spinal cord or brain, determining the appropriate response to the stimulus. Sends signal to effector. Once the information is processed, the sensory neuron sends an impulse to the motor neuron, which in turn sends signals to the effector, muscle or gland. Option C accurately represents this sequence, as the sensory neuron first receives stimuli, processes information, and then sends a signal to the effector to produce a rapid and involuntary response. Incorrect options. A. This option incorrectly places the motor neuron in the middle of the sequence, disrupting the correct order of events in a reflex arc. B. This option incorrectly suggests that the motor neuron receives stimuli, which is not the case in a reflex arc. D. This option incorrectly places the motor neuron at the beginning of the sequence, which is inconsistent with the typical reflex arc pathway. Therefore, option C is the correct answer, as it accurately reflects the sequence of events in a reflex arc. Embarking on the journey to become a healer is no small feat. Medical students face a rigorous path, filled with challenges that test their limits. It's a world where anxiety and pressure are constant companions, yet amidst the trials, a strong will emerges. This story isn't just about the struggle. It's a testament to the resilience and determination needed to push through doubts and fears. Join us as we explore the journey of a med student committed to saving lives, no matter the obstacles. Yo, it's the med student grind. Gotta stay in line. But my mind's like a jungle, it's hard to define. Books stacked high, but I'm feeling low. Pressure on my chest, like I'm moving slow. Every exam's a battle, gotta stay on track. But anxiety's a demon, it's holding me back. Stethoscope round my neck, but it's weighing me down. Feels like I'm drowning in this sea of renown. Late nights in the lab, coffee by my side. EKGs and charts, gotta take them in stride. But the fear's creeping in, like a silent thief. Got me questioning if I'll ever find relief. In the hustle and the bustle, gotta keep my head high. But anxiety's a shadow, it's hard to defy. In the world of medicine, where the stakes are high, I'm just a med student trying to touch the sky. White coat on, but inside I'm shaking. Got a mountain of knowledge, but the doubts keep breaking. Patients looking to me for answers and care, but inside I'm struggling, it's a silent affair. Cadavers in the lab, but they don't scare me. It's the thoughts in my mind that constantly dare me to doubt my abilities, to question my worth, in this cutthroat world where success is the girth. But I push through the pain, gotta rise above. Though the road is tough, gotta show some love. To the dream that drives me, to heal and to save, gotta keep the faith in this endless wave. In the hustle and the bustle, gotta keep my head high. But anxiety's a shadow, it's hard to defy. In the world of medicine, where the stakes are high, I'm just a med student trying to touch the sky. So I'll face my fears with every step I take. Though the ground may shake, I won't forsake. The dream that ignites me, keeps me alive. In this journey of medicine, I'll continue to thrive. Yo, it's the med student grind, but I'll make it through. With resilience and courage, I'll see it all through. Anxiety may knock, but I won't let it win. In the world of medicine, I'll find my kin, and then I'll find my gin. Meaningful scriptures from the Word of God to think about. Psalm 55, 12, 13 KJV. For it was not an enemy that reproached me, then I could have borne it. Neither was it he that hated me, that did magnify himself against me. Then I would have hid myself from him. But it was thou, a man mine equal, my guide, and mine acquaintance. Proverbs 7, 12-17. She was the brash, coarse type, seen often in the streets and markets, soliciting at every corner for men to be her lovers. Proverbs 29:11 ESV. A fool gives full vent to his spirit, but a wise man quietly holds it back. NIV. Fools give full vent to their rage, but the wise bring calm in the end. NASB. A fool always loses his temper, but a wise person holds it back.
Survival question alert. How do you feel about changing your diet to prevent disease? Which biological process or concept interested you the most in this video? Please comment below. How has learning about these topics impacted your understanding of human biology? Please comment below. Are there any other areas of biology you'd like me to explore in future videos? Please comment below. Hello there, I am Angelique Granville, and I'm a professional listener, digital products entrepreneur, and content creator. I have a degree in biology from Adelphi University, and I am passionate about helping allied health professionals succeed. I am here to help you get into medical school. On this channel, I share my tips and hacks on how you can pass your entrance exam so you can get into medical school. Do you want to talk to me for an hour about your life and your problems? Check out my therapy programs. The link is in the description. It says, "Let me help you in life." If you are getting value from this content, I urge you to like, subscribe, and share this video with all of the pre-medical students, biology students and majors, physics majors, and engineering majors that you know. If you know anyone who is struggling to study for the MCAT, please tell them about the Hospital Near Me Medical Career Training Channel. Together, we can conquer you getting into medical school and feel better about being accepted by the college of my choice. Please hit the notification bell so that you never ever miss another one of my videos. If you haven't already liked this video, make sure to give it a big thumbs up and send this video to at least 3 people. I know you can think of them. If somebody pops in your head right now and send it to them, WhatsApp, Facebook, TikTok, LinkedIn, and Instagram. Challenge them to do it with you and you can get results together. Come and take my easy MCAT exam. If you are getting value from this channel, all I ask is that you let my ads play on the video. And if you could bless my PayPal, Monzo, Cash App, Venmo, Buy Me a Coffee, GoFundMe, Patreon, or give me a thank you donation of $1 to $5, or with a donation of any amount, it would be greatly appreciated. I am saving up for a new home as my old one got severely damaged in Hurricane Sandy and left me homeless. I want to thank you for watching the Hospital Near Me Medical Career Channel for allied health professionals. I look forward to being a part of your medical school journey and being a part of your own growth. Walk with God. Thanks a bunch for watching this video. Thank you for joining us on this educational adventure. Don't forget to like, subscribe, and hit the notification bell for more intriguing content about the wonders of biology. Please like and subscribe to Hospital Near Me and tell your friends about this channel. Thank you for watching and stay tuned for the Shopping Network. Here is some useful information for you, and I quote, "Suicide warning signs for youth. Be a lifeline. It's time to act if you notice these signs in your family or friends. 1. Talking about or making plans for suicide. 2. Expressing hopelessness about the future. 3. Displaying severe, overwhelming emotional pain or distress. 4. Showing worrisome changes in behavior, particularly in combination with the warning signs above, including significant withdrawal from or changing social connections, situations. Changes in sleep, increased or decreased. Anger or hostility that seems out of character or out of context. Recent increased agitation or irritability. If you or someone you know is struggling or in crisis, help is available. Call or text or chat 988lifeline.org, or reach out to a mental health professional. Again, if you want to talk to me, please click on the link in the description that says, "Let me help you in life." Thank you for what you do.